I'm a plant physiologist, which means basically I'm interested in how plants work, how they function. But we do a very integrative approach to biology. So we start off being interested in plant function, and a lot of the people in the lab then try to work out what the genetic or molecular or biochemical basis is for that physiological performance. You know, why is it good in some conditions, bad in others? Why is it good in some varieties of a crop but not in others? So we spent a lot of time working in that space, and that's primarily what I'm going to talk about today. But we're also interested in how plant function scales up and upwards. And so if that would be the equivalent of, you know, me measuring your, say, respiration rate and then inferring something about your weight and your health and your life expectancy. But in the plant world, we would scale that physiological function to how much yield, how much food do the crops produce, how does their activity impact the carbon cycle. And we spend quite a bit of time in that portion of the lab group working with mathematical modelers and trying to represent that plant function mathematically, and then that feeds into models that are used to do things like project future food supply or project future climate change, because plants actually have the capacity to change the future trajectory of climate change quite considerably. So that's sort of a very broad view of what we're doing, but today I really want to talk about using genomic tools to understand more about how crops are going to respond to global environmental change, and today particularly the rising carbon dioxide concentrations in the atmosphere. So, probably all know this, but just want to reinforce the fact that the climate is changing in many ways. Increasing concentrations of greenhouse gases are, you know, the starting point here. We burn fossil fuels, we release greenhouse gases. We cut down forests, we release greenhouse gases. Those get into the atmosphere and cause warming. I've got it shaded in green here because, for reasons that I'll explain in a minute, higher carbon dioxide concentrations in the atmosphere per se actually help plants because they make the photosynthesis process more efficient. But obviously when we step back and look at the bigger picture, that rising carbon dioxide concentration in the atmosphere is really bad because it triggers all this other stuff. It causes warming, and that's a mixed color here because things getting warmer, that's good or bad depending what temperature you start off at, right? So in the Midwest, it's already pretty warm during field season when the plants are growing, so getting warmer generally a bad thing. I'm originally from Scotland. We'd be kind of excited if it got a little warmer. And it probably wouldn't hurt our crops too much. And so, unfortunately, I should probably change where the dividing line is, and it shouldn't be 50-50. A little bit of it should be green and most of it should be red, but there is a geographic context there. As a function of the overall climate warming, we accelerate the hydrological cycle, we accelerate the water cycle, and that manages to do this unfortunate combination of increasing flooding and increasing drought. And that's partly a product of making the dry places drier and the wet places wetter. It's also partly a product of getting more rain in big storm events. So you get a big dump where you get more than I mean, last week would be a beautiful example of that kind of event. Can't say that it was linked to climate change per se, but you know, that sort of big rain event, not very helpful if you're trying to grow crops, interspersed by longer periods with no rainfall. And then one other one that's slightly related to these but somewhat separate is we're also really interested in tropospheric ozone, and so that's the primary constituent of smog. If you've ever flown into LA and seen that brown layer, if any of you have asthma and you've got out there and you feel terrible, it's because you're breathing in ozone and it's causing damage to your lung tissue. If you're a plant sitting out in the field, you're breathing it in too, and it causes major problems as well. So those are all different abiotic facets of climate change that are occurring and that plants have to deal with in the future. And so we've got a problem where we need to feed ourselves, and we need to try to do it more sustainably. And so we need to try to maximize our future yields and our efficient use of the landscape. And in order to do that, we need to try to mitigate all the negatives. So all the red things here, we need to try to reduce the degree to which they're negatively impacting the plants. And where we have these little blips of green, we need to try to maximize those benefits. And it's this CO2 one that I'm going to mainly talk about today. Just to give the whole climate change thing some context for you, I find this to be a very compelling way of describing what the future will look like. And so this is data based on some people who are at the Department of Atmospheric Sciences here at Illinois. They basically looked at the projected climate for the end of the century, and they looked across the US at climate variation now, and they determined that what Illinois will experience in 2095, the closest proxy for that is what Eastern Texas is like right now. So that's what our future might look like. We're moving on that sort of a trajectory.
I'm actually from New Mexico. Does that mean I'm headed towards Mexico City? Does this work across all longitudes?
I mean, this is a bit of a simplification. So it's not a question. It's not. They've made it look here as if the whole of the Midwest is slipping in that direction, and that approximately that's kind of true. Yeah. But it's not a perfect description of exactly what's going to happen, but it gives you some sense of where you are. It wouldn't be a million miles off, but. And so clearly, for those of you who visited Eastern Texas, they don't grow a whole lot of corn and soybeans there. Life's going to be tough to maintain the same agricultural systems we have right now. And so we're going to have to both try to slow the rate at which we're changing the climate and adapt to the change, to the certain amount of change we're guaranteed to get. And that's where we come in. I wanted to quickly show you the change in CO2 concentrations that have been directly measured since 1958. A guy called Keeling decided in 1958 it'd be a really good idea to actually be trying to directly measure how much CO2 is in the atmosphere. And he went to the top of a volcano, Mauna Loa in Hawaii, decided that would be a good place to do it. Presumably some of that was based on the fact that Hawaii is a cool place to be, but as researchers we do that stuff when we can. But his primary motivation was that was a big volcano up in the atmosphere, high in the atmosphere, a long way away from any major sources of industrial activity. So it was giving him a pretty good measure of sort of a global average CO2 concentration. And when he started out in 1958, he was getting values of 320. So depending on how familiar you are about thinking of parts per million as in terms of gas concentrations, what that's telling us is that when he went to the top of his volcano in 1958, if he had been able to grab 1 million molecules out of the atmosphere, 320 of them would have been CO2, right? So it's less than 0.04%. The vast majority of what's up there is nitrogen and oxygen, but there were 320 parts per million of CO2. Now in 2014, we've actually, 2 months ago was the first month where it stayed above 400 ppm all month in about 800,000 years. So that was a major threshold for us to pass, and the projection is for that to keep increasing increasingly rapidly as we go forward. So we're looking at concentrations of probably somewhere around 500, 600, 700 by the middle of the century. That's definitely not occurred on Earth for a very long time. But if we think about this in terms of crops and agriculture and the ways in which we play a role in developing those crops and trying to make better crops, we've actually been, drought and temperature and flooding, we've been dealing with those for a really long time. So modern agriculture sort of got going 7,000 or 8,000 years ago, somewhere in the Middle East. And since then, people have been out selecting the varieties that do better in whatever their local conditions are. And if you happen to be if you've been farming in the Middle East for 6,000 years, you've probably managed to find some varieties that do pretty well with drought. And because we've been so conscious of the challenges that those present, and that those have been such long-term challenges, ever since we've been using modern breeding and biotechnology approaches to improve crops, we've been trying to tackle these things. I mean, if you go to Monsanto and Syngenta and places like that and say, hey, I'm interested in drought, they all nod enthusiastically because they're interested in it too. What they haven't, and if you look at the products that are coming out of their research and development pipelines, Monsanto released a genetically modified corn plant a year or two ago, which is designed to do better under drought conditions. So these are things they've been, and that's a slow process. That if they've released that last year, they started that process 15, 20 years ago. It doesn't just pop out. That's a very slow progression. But we haven't done the same thing for high CO2. We've only really appreciated that it's going up since the 1960s, and and most of the research that's been done to study how plants are responding to it has been done in the last 30 years. And certainly, we get invited to go down to the seed companies every so often because they now have this CO2 idea on their radar, but they haven't done anything about it yet. It's a far enough away target and a new enough idea for them. It's not something they've got to grips with. And so we're trying to do the real sort of upstream research that will feed them towards trying to tackle this sort of problem. And so what we're interested in is, can we develop the knowledge that would allow us to use biotechnology or breeding to improve crop performance for the CO2 concentrations that are going to occur in the future? And that's important because this map shows you a pretty optimistic estimate of how yields might change by the end of the century as a consequence of climate change. And so these are percentage changes in yields in 2080 compared to 2000. And so anything that's a hot color is a decrease in yield. And so what that's telling you is that those increases in temperature and those increases in drought, stress, and flooding, they're having a pretty negative impact. And the small positive effects of CO2 aren't doing enough to counteract that. And we want to see if we can get more benefit out of the CO2 element. The main way in which CO2 actually benefits plants is it makes photosynthesis more efficient. So if we take a soybean that's growing in today's CO2 concentrations, and we refer to that as ambient CO2, and you compare that to a soybean that's growing in the CO2 concentrations we expect for the middle of the century, operating in elevated CO2 really makes Rubisco, which is the primary enzyme that actually captures the CO2, makes it work a lot more efficiently. And so it captures much more CO2 by photosynthesis and converts it into more sugars. Rubisco turns out to be the world's most important enzyme, but also one of the more stupid enzymes in the world, because it sometimes accidentally confuses carbon dioxide for oxygen. And instead of grabbing a carbon dioxide and adding it to a sugar, it grabs an oxygen and adds it to a sugar and makes a product that's actually toxic to itself. Pretty dumb all round. It does a very clever bit of chemistry, so I guess it's a little unfair for me to call it stupid, but it does make mistakes. And that mistake is called photorespiration, and it really hurts the plant. And elevated CO2 actually counteracts that. Just the fact that you've got more CO2 in the atmosphere means that it has less opportunities to screw up and accidentally capture an oxygen. And so the net result of that is these plants make more sugar, the guy on the right is bigger, and a bigger plant will generally generate more yield. So we get about 15% more seeds, on the weigh scale at the end of the season per acre of land when we grow soybeans at the CO2 concentrations expected for the middle of the century. You have a question, Barbara?
Yeah, so you said something, you'll come to later, tolerant seeds are not going out. Is there any correlation in the biochemical pathways between making something drought tolerant and something that is more capable of living in the
That's a good question. Potentially, for reasons that I haven't really got in this slide deck. But the honest answer right now is we don't know. That was a very sensible question to ask, and there are some reasons to think it might be true, but we don't have the data to say one way or the other at the minute. Yes?
Just because we're here, can you go back a slide?
Oh, sure.
I was just, okay, so this is, what is this showing us?
This is showing us how yields would change in 2080 versus 2000.
Okay.
So if
But
you're...
it's nothing to do with the CO2? Right?
Yes.
It does, okay.
Yes, so this is the net effect of the negative effects of temperature, the negative effects of drought, the negative effects of flooding, and the positive effects of elevated CO2. So you're sort of mashing all those things together in a mathematical model. Actually, this is the output of a number of mathematical models that have then been averaged. So this is our sort of, it was, when it was published in 2007, our best guess of what was going to happen in the future.
So the idea is that if you somehow make plants grow more efficiently, that map might change.
Yes. So if we can get plants to benefit more from high CO2, there would be less red on this map and maybe some more green and some more blue. Yeah. Thank you for asking.
I just want the name of the journal. It's Klein?
The book is by a guy called Klein.
Klein.
Yeah. I've already given them the PowerPoint slides, so if you want stuff out of the slides, I'm sure you can take them.
On the other one, you said that, is there anybody working to genetically fix stupid Rubisco enzyme to not bind?
Oh, yeah, that's a great question. Yeah, loads of people are doing it. Turns out to be a total nightmare. People have been trying that for at least a generation. Rubisco does it makes mistakes, but it does a spectacularly difficult piece of chemistry. It does it with an incredibly complicated protein. So it is a, let me think, what's the proper word? I want to say it's a diheterooctamer or something. It's got 16 subunits. Eight of one. So there's two bits of it. There's 8 subunits of one protein, and 8 of the small. It's called the small subunit, and then 8 components of the large subunit. And so you have to. It's like the world's worst bit of Lego. You've got to be able to glue all 16 bits of protein together in the right place in order for the active site of the enzyme to operate properly. And so it's very difficult to go in and monkey around with something that will make it perform better and not just mess the whole thing up. And that's made even more difficult because the large subunit is encoded. Oh, actually, which way around is it? Can't think right now. One of the subunits is encoded in the nuclear DNA, where most of our genes reside. But if you're a plant, you've also got some DNA in your chloroplast, so sort of separate to your regular chromosomes. And the other subunit is encoded in the chloroplast DNA. And so in order to actually make this enzyme, you need gene expression in the nucleus and in the chloroplast to both happen in sync. You need the proteins to get made out in the cytosol, and then you need them to all appear from those different sources in the right place in the chloroplast and glue together right.
So the fact that it even works at all, they're willing to kind of just say, that just makes it an incredibly difficult thing to poke and have work at the other end. You just tend to knock the whole thing down. And so as a genetic engineering target, it's particularly difficult. But if we could fix that, that would solve an awful lot of our problems. Interestingly, there is quite a bit of genetic variation in Rubisco, and so lots of people are out now dashing around the world looking at the performance of Rubisco from lots and lots of different photosynthetic organisms, from algae, red algae that live in volcanic mud pools, through to seaweed, corn, soybean, every different type of photosynthetic plant, cacti, and everyone's out there sort of almost bioprospecting, trying to find a plant that happens to have a Rubisco that's a lot better. Interestingly, evolution seems to have done about as well as it can in all of those settings, so there's another really terrible trade-off if you're a Rubisco, which is when people look across all of that variation, if you have a Rubisco that's really good at not screwing up and combining with oxygen instead of CO2, it tends to do its reaction really slowly. So, it's a total plodder. It gets the answer right, but it gets there really slowly. If you have a Rubisco that goes really quick, it screws up more often. And so you when you try to improve one, you tend to break the other. It's a really interesting bit of sort of constrained evolution where Rubiscos have been made along this sort of axis of trade-off and haven't managed to get a lot better. But it's if we could crack that one, we could bump up crop yields to a spectacular degree probably pretty darn quickly. And another little factoid on Rubisco, because it doesn't work very well, it's the most abundant protein on Earth. So plants have to make lots and lots and lots of it in order to do photosynthesis. About a quarter of all the protein in every leaf of every plant on the planet is Rubisco. So there's way, 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 way more Rubisco than anything else. I mean, orders of magnitude more of it than the hemoglobin that's in our blood, that sort of stuff. So it's a pretty amazing little protein, or big protein, I guess. So the bottom line here is high CO2 can sort of be a silver lining to climate change, if you like. It's unfortunately not as thick a silver lining as we would like it to be. And we've been quite interested in whether maybe we've been accidentally improving the situation as we've gone through, because crop breeders are out there every year growing lots of different varieties of crops, crossing lots of different varieties of crops, getting novel combinations, and then picking the best ones. And lots of people have said, oh, well, maybe we're actually accidentally, because CO2's been rising over the century, maybe we've been accidentally picking plants that are better at taking advantage of that high CO2. And we haven't got a huge amount of data on that, but it seems like that's not the case, and some of the data sets that have been collected suggest that maybe it's even going in the opposite direction, which would be really bad. And so one of those data sets is from a group in Maryland, and what they did was grow some wheat at a bunch of different CO2 concentrations. So down here at the bottom, so we've got 4 varieties of wheat,
A, B, C, and D. And those 4 varieties of wheat were released by wheat breeders. So when wheat breeders come up with a new type of wheat that they think is really good, they put it out in the market. They call that releasing it. And so the earliest one of these was released in 1903. So 1903, wheat breeders thought that was a great variety that people should be growing. In 1921, they released another one called Thatcher. 1965, another one called Chris, and in 1996, another one called Oxen. Now, this is just 4 out of hundreds of varieties of wheat that have been released over time. But the question that Zizka and his buddies wanted to ask was, is that one that was released recently better at doing well in high CO2 than the other one? And it's actually gone the wrong way. So if you look at the performance in 700 parts per million of CO2, which is, so this might be how much yield you would get per plant towards the middle or end of the century, you can see that's been going in the wrong direction, at least for those 4 particular varieties. Now, that's not a particularly comprehensive test of that question, but it means we might not have really been helping ourselves. So this encourages us that we've got to put more effort into tackling this question.
So why is there no yield at 600? Is it that the plants died, or because they didn't?
Oh, they only did the experiment at, what is it? That's probably 350, 400, and 700. 750, yeah. That's just the conditions they had available to them. And so lots of us here at Illinois are interested in this. In fact, I moved here 12 years ago because this is probably the best place on the planet in terms of people thinking about this question. And so we sit around and think about this, and we're pretty convinced that because we understand how plants respond to high CO2, we can come up with some ideas of things we can manipulate to try to improve the situation. Oh, here's a picture of Rubisco. So this gives you some sense of how complicated a protein it is. The light blue and the dark blue are one subunit, and then the reds at the top are another. So it's this weird sort of barrel-shaped thing with a whole bunch of bits. And so we spend time from first principles coming up with ideas of things we might change.
Why is this the best place?
Just the coincidence of who happens to be here.
People.
It's all about the people. Yep. Plus the facility that I'm going to tell you about in a minute. But that was a product of the people, I guess. Yeah, we have some tools here that other people don't have that I'll show you in a second. So we can do some stuff this way, but that's constraining us to the things we understand. And there's way more things we don't understand than we do understand. And so that's where the sort of genomic revolution has come in and allowed us to take a new approach. And there's a couple of things I want to remind you about this and explain why we looked at the changes in gene expression that occurred in plants at high CO2. One of them is that many key adaptations are the results of changes in gene regulation rather than gene content. And so a classic example is humans versus chimps. Our genomes are 99% the same in terms of the content of the genes. But at least I think we like to think we're at least more than 1% different than chimps, right? The argument here is that there's a few key genes, those master genes that are good at switching on and off lots of other genes, and we call those transcription factors. And there's a few of them that are very different between us and chimps. And so that's a change in the way our genes are regulated rather than a wholesale change in the sort of set of genes we have to play with to begin with. And so understanding which genes get switched on and off when and what the other genes are that regulate that process can really help you understand biological diversity. And so that's one step. The other thing is that process of altering gene expression is also one of the key ways you can change the physiological function of an organism. Or you can, they also affects the process we call acclimation. And so one of the best examples of acclimation that people know from the human world is if you know any Olympic athletes who are going to go to Rio next year, you can pretty much guarantee that they're going to go live at high altitudes for a couple of months before they go to Rio, right? And the reason for that is that they're intentionally tweaking their physiological performance by acclimating to the low oxygen concentrations you get at high altitude. And when you take a human body and you put it in low oxygen, it reacts, it senses the fact that it's not getting enough oxygen to the muscles and brain and everywhere else, and it reacts by switching on the genes that make red blood cells, and therefore increasing the efficiency of oxygen transport. And these guys here have published some nice papers where they actually understand the genes that switch that process on and off. And so what we would like to do is a similar sort of thing, where if we grow plants at high CO2, can we work out the genes that the plants switch on and off in order to perform better in high CO2? And this kind of summarizes the approach a little bit. And so because we're physiologists and because we're integrative, this is sort of the overall roadmap for the way we think about things working. Some aspect of the environment changes, in this case CO2 concentration in the atmosphere. And the red arrows represent the fact that that has direct impacts on various molecular, biochemical, and physiological processes. And then those all scale up in order to and combine to somehow impact whole plant performance. So that would be growth, yield. That alters the way the canopy performs. So it actually changes, for instance, how much water the canopy uses. That might alter rainfall in an ecosystem context, but it's also going to feed back and influence what soil conditions are like and how that plant's going to experience life later in the growing season. So we have to sort of put together this whole network of things across all the different levels of biology. And so I think traditionally, if you go back a generation, biology was still thought of in a relatively compartmentalized way. If you visited universities, there was a department of cell biology, there was a department of organismal biology, there was a department of ecology. And what's changed a lot, particularly in the last decade, is there's now lots and lots of us who try to think about, well, how do you see the way all those different levels interact with one another? And if you actually want to solve real world problems with biology, we've discovered that if you ignore this, you're pretty much dead in the water. Because something that changes up here is often counteracted down here. So this idea of being able to see that network is pretty important. And so the technology that we have that's come along in the last 10 or 15 years are things like, have you heard about RNA-seq this week? Yeah, so RNA-seq is the latest way of looking at gene expression. When we were doing these experiments 8 or 9 years ago, we were using a thing called microarrays, which also allowed us to look at gene expression. And so that was technology that suddenly gave us a whole bunch of new information of what was happening here. At the same time, we can do high throughput biochemistry, where we can look at a lot of the individual metabolites in the plant, and that's just represented down here. So for a long time, we were trying to answer this question without being able to see what was happening here. And that made life really difficult, because that's where a lot of the primary events were occurring. And that's what we're trying to tackle here. So it sounds like you've done lots of stuff on the central dogma of molecular biology this week, so I'll pretty much skip this, other than to say, you have DNA, a copy of which gets made into RNA, which then gets read and turned into protein, and it's the expression of proteins in different cells that leads this to be a leaf versus this to be a root. Or in the case we're going to look at, it's going to be the gene expression altering the protein content of plants at ambient CO2 versus elevated CO2, which is going to change the way they behave. Have you talked about microarrays, or should I explain that? Okay, so this is slightly old technology still, although we still use them actually, because the data analysis involved in doing the RNA-seq you learn about is so horrific that sometimes it's just quicker and easier to do this. And you can learn just as much a bit faster. Essentially what you do is you take all the RNA out of the bit of tissue you're interested in, and then you wash it across this array. And so they look like this. This is about just over 1 centimeter in length, and this is it zoomed in. And the manufacturers have gone in and they've put a little strand of DNA in dots all over this. So every one of these little spots is a little cluster of strands of DNA that corresponds to just a portion of the RNA that might be out there. So you could think of it almost like it's not a dot per gene, but you could kind of think of it that way. There's a dot that represents the DNA sequence for every gene that might get expressed, or close to every gene that might get expressed. And then you can basically make a DNA strand from your RNA, which is going to be complementary to the one that's on the chip. And then essentially your RNA is then just washing across the chip, and when it comes to its matching buddy, it binds to it. And you've added a little fluorescent tag on top of it. And so if I have 10 copies of my alcohol dehydrogenase gene floating around in my liver because I had a beer last night, those 10 copies would wash around until they bumped into the spot on the slide where the alcohol dehydrogenase gene is, and then you'd get a little fluorescent
blip because 10 of those little fluorescent tags were visible there. And some other gene that wasn't expressed last night is going to be a dark patch. So essentially you can see which genes are switched on by using a microscope to look at which are the bright spots and which are the dark spots. You get a quantification of how much RNA you had for close to every gene that was in those cells.
Sorry.
Do you want to clarify any of that?
Yeah. So on the array, the manufacturer has just put a genome, or what? I don't understand the ground.
So they're cDNAs. It's half of a cDNA. So
do you specify what you want on the chip?
It's close to a whole genome. It's very close to a whole genome. Essentially.
There's still a cDNA?
So they made them from cDNA libraries. So they would go in. This was before the soybean genome was sequenced, but they would go. They tried to get unique identifiers, and so there's 38,000 probes. We call each of those spots probe, and so we essentially treat it as a whole genome array.
And then you put your express sequences over it.
Yes.
And then how do you identify your alcohol and dehydrogenase?
Because they essentially give you a map of where each of those genes is located physically on the array.
So this only works for genomes that have been sequenced?
Well, it's not fully sequenced. You have some sequence information because they actually artificially synthesize the probe sets, and so they give you that information. So you're getting a 20mer, and then you have to do blast searches to just see how that matches up. Does that make sense? Mm-hmm. So this would be just another representation of it. If you were to zoom right in on one of these dots, this would be a synthetic oligonucleotide, synthetic set of DNA bases that the manufacturer has made, and they might know that ATC, ATG, is a unique stretch, in a given gene, and then if we have the complementary strand that matches that one, it's going to come along and bind, and you're going to see all those because there's going to be a little forest of them in a cluster, and then that's going to come out like a bright spot. Does that be hideously confused you? No, everyone's nodding. That's good. So in essence, we're just getting a quantification of how much mRNA there is there for every gene, or close to every gene in the genome. And so we wanted to do experiments to identify which genes were changing in plants growing at high CO2. That just hadn't been done before. And we particularly focused on leaves, because leaves are where a lot of the action is. They're doing all the extra photosynthesis, plus some other stuff we're going to talk about in a second. We did this in soybean for two reasons. One, it's the fourth most important crop in the world in terms of just total yield, so people care how it's going to respond to high CO2. But also because we have a whole bunch of it in our backyard. We're here. This is the USDA map of soybean acres planted, and you can see we're slap dab in the middle of the major growing region, so this is a good place to do it environmentally. And then from just a straight science point of view, it's a good model system to work with. The Midwest is about as environmentally uniform a place to do an experiment as you can find, because we wanted to do this outdoors, as I'll show you in a second. Soybeans great because of very low genetic variability compared to some other plants. Pretty much every soybean plant in my field is genetically identical, so that's not going to mess with my ability to do a good experiment. That gives me a lot of statistical power to detect treatment effects. If I'm going to do an experiment, I'll tell you about an experiment in a second that had 4 replicates. So I had 4 control sets of plants and 4 elevated CO2 sets of plants. That's not a massive amount of replication to try to find differences, and so having a nice system is good. The other thing is, at the time we were doing this and genomic tools were not as available as they are now, microarrays were available for most of the major crop species, so we could do this sort of stuff. And then soybeans actually a pretty easy thing to come up and measure. It's got nice big leaves, you can do stuff to it relatively easily. And compared to what our ecological colleagues were up to, every plant's the same, they're not competing with different species or doing anything complicated like that. This is the technology we actually use to do the experiment, and this is the other thing that Barbara was alluding to earlier about why I would come here. We have probably the biggest facility in the world for growing plants outdoors in a farmer's field, but with conditions that simulate the future. So we have an 80-acre field site near the airport where we can grow plants at high CO2 or high ozone or high temperature or under drought stress, and do it in the field, not in the artificial conditions of a glasshouse. And the technology we use to fumigate the plants with CO2 is called FACE, stands for Free Air Concentration Enrichment. And so if we have, this is a photograph of just one of our plots, and so they're about 20 meters in diameter. It's got an octagon around it. If you imagine, the green pipes release the CO2. If you imagine the wind is coming in the direction of this arrow, there's a weather station in the center of the plot that measures the wind speed and wind direction. It sends information back to a central computer every 4 seconds, and that computer then opens valves on this box that release CO2 from the 3 upwind sides of the ring, and CO2 gets carried across and through the plot. And then that little white box right in the middle is a CO2 analyzer that sets up a feedback loop. So if the CO2 is below the target we want to hit, it releases more. If it's above, it releases less. It does a quite substantially fancier bit of math than that. In order to, it's called a predictive algorithm. So if you just did high, low, on, off, it would tend to overshoot and then undershoot. And the predictive algorithm does the math so that it's sort of accounting for what it did 5 minutes ago in what it does now, to sort of hit a level turn. And if it changes wind direction, it just releases CO2 from a different side. Yes?
Do you have a way to monitor within that perimeter, like the CO2 concentrations for the different locations within there?
That's a great question. Yes. We don't do that routinely because it's expensive and it takes a lot of time. But when we go across, but we have done it several times, and when you go across the space over a whole season, it averages out. So at any moment in time, you have a concentration gradient where it's highest near the release point and lowest further away. That concentration gradient is steeper when the wind speed is low. When winds are relatively high, it's not such a problem. And we get quite high wind speeds here, so that helps us. But yeah, there's enough variation in terms of coming from different directions that it all averages out over the course of a season. Except for right next to the pipes. And we basically have a 1 meter perimeter zone that we don't go anywhere near right next to the pipes because they get crazily high concentrations.
So there's been no, I'm just wondering, would like a biodome work better?
Oh, so people did that before. So the traditional technology has been to use some sort of enclosure and pump CO2 in. We still do some of that kind of work, and we have cabinets in the building here that you can walk into a room, and they have lights and temperature control and CO2 fumigation. And that is useful in certain ways. The problem is the temperature is different, the light's different, the moisture is different. Usually the amount of space the crop has to grow is really highly constrained. What we've discovered is that when you don't do it in the field, you tend to get really different answers. And so actually going and doing it in the environment that you want to know about in the future is a really important component of this. So when I said that map earlier was very optimistic, the color-coded one of how yields change in the future, that map was made when we thought that yields would increase about 30% in the CO2 concentrations predicted for the middle of the century. That data was based on people using that kind of approach. When we and a network of other people around the world went and did this in the field, we got a number about half that big. So we published a paper now 8 or 9 years ago that got a huge amount of press coverage because basically we were arguing that that map was way too optimistic and it should be even redder than it was now that we'd kind of gone and done the experiment out in the real world.
Just coming back to that map real quick, I mean, that was just on CO2 concentration.
That was on CO2, temperature, drought, all combined.
You notice the northern latitudes in their growing season extends, just like you said, Scotland would benefit from that stuff. So
yeah.
That was a big picture.
That was a sort of an amalgamation of everything.
And then inside a controlled, like a room, are you messing with actual partial pressures of the gases? And does that affect, and that might be a reason for the differences in, I mean, if you're pumping more CO2 into a chamber as opposed to this sort of ambient environment, I mean, does partial pressure
Oh, we're not pressurizing the chamber to a meaningful extent.
Here,
So,
you're actually
yeah.
controlling for that.
No, yeah, no. That would actually be really difficult to do.
Yeah.
Part of it's because CO2 is such a small fraction of the total concentration that you're only dribbling a tiny bit in terms of volume. So, but yeah, no, that's a good question. I need to be careful of the time here because we've barely started, and so I'm gonna crash on here a bit. So this is just very quickly the experimental design. So I said we have lots of these rings. We actually have 32 of them altogether between corn and soybean. But in the experiment I'm going to describe to you just now, we had 4 of them that were under ambient CO2 concentrations, which back when we did this, it was only 385 instead of 400. And we had 4 of them that were in elevated CO2 at 550 parts per million. And basically, we build all that infrastructure on top of the crop right after after the tractors go through planting, and we switch it on by the time the plants start emerging out of the ground, and we fumigate them from dawn till dusk every day from the beginning to the end of the growing season.
Why only dark dusk?
We don't do it at night for 2 reasons. One of them is that high CO2 when the plants are in the dark doesn't do anything. Although to be totally honest, is more a matter of convenience. At night, CO2 is really expensive. We spend about $50,000 or $60,000 a year on CO2. And so if we can we would have to double that bill if we get at night. And I mentioned wind speed earlier. Winds at night are often really low. And so the system works terribly at night a lot of the time. And then the third part of it is that CO2 concentrations at night are often naturally really high anyway. So if you get low wind speed, all of the respiration that's occurring from the plants in the soil, all that CO2 sort of gets stuck in the canopy, and you get really high CO2 concentrations anyway. So when that happens, we then have a really hard time deciding what we were trying to fumigate to. So when you put those 3 things together, it just doesn't make any sense. In the early years, we ran it at night to see if we could create an effect, and we couldn't. Yes?
How do you determine that you would use 550 parts per million? Is that what the projected level will be in your...
Yeah, that was like a best guess for 2050.
Okay.
Yeah, good question. And so basically what we did was then in 2 successive years we went out and we took leaves that were sitting at the top of the canopy at 4 different developmental stages through the season. So full bloom would have been occurring probably around about now, and then the week after that, the week after that, the week after that, we keep walking out to the canopy, find the leaves at the top at midday, and then collect some of those, put them in little tinfoil packets, plunge them in buckets of liquid nitrogen that we walk around with, and then bring them back in to the lab. So it's all different leaves, but this is just showing you what the plant was doing at the time that we picked those leaves. And so we focused on the period when the plants were actually filling the beans and growing seeds. This was just to remind you that we said high CO2 stimulates photosynthesis. But our question here was like, well, what are the changes in gene expression that occur alongside that, which is the plant sort of tweaking its setup in order to perform best, kind of the equivalent to those Olympic athletes going to low oxygen. And we obviously had a hypothesis. Our hypothesis was that you're going to get the maximum efficiency out of the system when that improved performance of the chloroplasts doing more photosynthesis is matched by changes and improvements in the mitochondria that are doing the respiration, because plants respire just like us. They need to do that. And so what that led us to predict was that there's going to be more expression of the genes that make the respiratory machinery, so that it can take advantage of all the extra sugar that the photosynthetic machinery is making for it. I don't know how much, when you've been talking about RNA-seq and stuff, you've been talking about the data side of things. This is a really fantastic example of why effectively training people in math is a really key component to generating good biologists. The data side of most genomic technology now is when 90% of the work and effort and struggle and difficulty is. And so when I give this as a seminar at a conference or something, we tend to joke about the idea that the statistics of doing this are like reading tea leaves. And I like to tell a story about this lady called Muriel Bristol, and who was buddies with, probably the most famous statistician of all time, Fisher, the guy who came up with the t-test, amongst many other things. And she sounded like quite a character, and she was a very fussy British lady who claimed that if you put the made to her milk by putting the tea in made her tea by putting the milk in first versus the tea in first. It didn't taste right, and she would tell you to take it away. This is a sort of claim to social status. And Fisher basically thought she was full of shit and tried to call her out on this. And so he actually created a whole new statistical test to catch her out. It's called the exact test, and we used it in doing this experiment. So I'll describe that in a second. And so we basically came in and did a... Oh.
So could she?
No, she couldn't. No. Yeah, he proved she was full of it. So you've got to be careful how smart your friends are, I guess, is the question here. And so we did. So we started off. If we go back to the array is capable of detecting about 37,000 genes expressed in soybean. There are lots of genes in soybeans. Plants are interesting because they tend to have bigger genomes than humans and other stuff. And when we did those initial statistical tests, when we averaged over the whole growing season, we got somewhere between 2,000 and 3,000 genes that came back as being statistically significant. We then took all of we looked up the bioinformatics for all of those genes and looked up what the predicted function of all 37,000 of those genes were, and then basically put them into 35 families. And so if you turn sideways, these are the 35 families. And so, you know, these were genes that might be related to making the respiratory machinery, or making the proteins that make the cell wall, or making proteins that are known to respond to stress, or making proteins that we don't know what's going on, so they're called miscellaneous. That's actually the biggest category. Making genes that make proteins we know are key in regulating development, protein synthesis, whatever it happens to be. So you put them in these groups. And then I looked and said, well, in each of those groups, what percentage of the genes were responding to high CO2? And that's what's on the y-axis here. And then I did that for those 2 successive years. And then I just ranked these by the average. So families of genes with functions, the related functions over here, were proportionally more responsive to high CO2 than the ones over here. And if you just took an average over the entire set, you know, we're getting around about 10% of the genes that are expressed in this tissue coming back as responding to high CO2. The question is, you know, for how many of them is that real? So if you think back to statistical tests, you know, and everyone obsesses about that p equals 0.05 thing, you know, when you do that test, you're saying, I think there's a less than 5% probability that the difference between these two conditions occurred by chance. And so that's a small enough probability that I'm happy I'm going to take it. The problem is, if you do that test 100 times, on 5 of the occasions, it's going to give you the wrong answer, right? And here we had to do it 27,000 times. So we're going to have a bunch of times where it gives you the wrong answer, gives you a false positive response. And so one of the interesting things about getting into this sort of technology is you have to, it's really the first time as a plant biologist I had to deal with that problem, because the data was getting big enough. And so you then have to identify ways of fixing this. And Fisher's test is exact test is one way of doing that. So it basically looks at one of these families and says, is the difference between that family and the global average statistically significant? And what we found was that, conveniently, because we had a hypothesis about it, one of the major families of genes for which CO2 was having a significant effect was respiration, and another major family related to this is carbohydrate metabolism, which is very tightly intertwined with respiration. So once we had this, that then encouraged us to go and look at the genes that are involved in those things in more detail. And that's effectively what I'm going to show you now.
So were those all upregulated? Or?
No, that was just any change.
Oh, any change.
Any change. And so we then wanted to go look at those pathways more, and because they're nice metabolic pathways, we can actually look at them as pathways. And so I'm just going to orient you with what I'm going to look at here. So metabolite pools are green boxes. So fructose 1,6-bisphosphate is a sugar with 6 carbons and a phosphate stuck on the end. Arrows are fluxes, where one metabolite gets converted to another metabolite by an enzyme. The boxes tell you how the expression of the genes changed, the genes encoding that enzyme. And sometimes there's more than one gene for a given enzyme. And occasionally you have a complicated situation like this one where there's actually 2 enzymes that are capable of converting fructose 6-phosphate into fructose 1,6-bisphosphate. So then you get 2 clusters of boxes. And then the colors just tell you whether there's more of a more gene expression in ambient versus elevated CO2. So blue boxes is turning genes up in elevated CO2. Yellow boxes is turning genes down. You don't need to be able to read any of this. This is just me reminding you, showing you all the pathways we're going to look at. So we're going to start off looking at the pathways that do photosynthesis in the chloroplast. This is the chloroplast. Then that makes sugar, and then we're basically going to follow the respiratory pathway that sugar enters as it goes through glycolysis, then gets shunted into the mitochondria, does the TCA cycle, and then does the electron transport where oxygen gets consumed. So we're going to sort of break that down. So here, if we look inside the chloroplast, if we think about photosynthesis, so 2 major chunks to photosynthesis. The light reactions, where light energy actually gets captured and converted into a chemical form. And then what we call the dark reactions here, where that chemical energy is used to capture carbon dioxide and make sugars. So this is the light reactions up here, this is the dark reactions over here. You can see there was a bunch of yellow boxes. So there's actually less gene expression for the photosynthetic machinery at high CO2. And that was the one part of this story that we knew about before. And that happens because the machinery is working so much more efficiently, the plant actually reduces investment in that machinery in order to free up some resources to invest more in other parts of plant performance. Because balance is really key here. You have one element is working great, but you've got a bottleneck somewhere else. Your overall process is not as efficient. But what we didn't know about was what happened after that. And this is where sugars are getting kicked out of photosynthesis and then being converted into starch. So this is representing starch granules over here. So fructose gets converted, fructose 6-phosphate gets converted into a couple of other sugars, then it gets made into starch. The plant holds onto that starch till nighttime, and it breaks that starch down and exports it out of the leaf to the rest of the plant. That's how it's feeding the rest of the plant. And so you can see that, I'll go up here first, in high CO2, there's actually more photosynthesis going on than in ambient CO2. That's just reinforcing what I told you right at the beginning. That results in more starch storage, and it's no coincidence we've got greater expression of the genes that make the enzymes that actually make the starch. So we can sort of see the biochemistry and the molecular biology fitting together then. The other thing we saw was that there was quite significant upregulation of some of protein transporters that actually allow the sugars to escape out through the membrane of the chloroplast into the cytosol. And so then if we follow that sugar out into the cytosol, this is a slightly complicated version of the glycolysis pathway you might be familiar with from textbooks. Doesn't really matter what any of the details are. The key point here is there's loads of blue boxes, right? So essentially all of the enzymes involved in glycolysis, their genes are being expressed more. And so we think that means, well, we're assuming, and this is admittedly a big assumption, we're assuming that means that there's more protein being made for those enzymes, and therefore those enzymes are able to be more active and push more material through the glycolytic pathway. And fitting that as well, we also looked at the key pools in here, so glucose, fructose, and sucrose are the 3 sugars in this pathway that are there in greatest abundance. And we went in and measured those, and then if we add those up, you can see that there was more of those pools in there. And so again, that's not totally watertight evidence on its own, but when you sort of add all these things up together, it starts to become convincing. Finally, if we go into the mitochondria, you'll see again tons of blue boxes. So it looks like basically every enzyme in this whole pathway is getting its genes turned on more in order to make more of the pathway. And then finally, we're able to actually measure the flux going all the way through that pathway by measuring the amount of CO2 that gets released but in the respiratory process, and the amount of oxygen that's getting consumed. So just like when we breathe in oxygen, we exhale CO2. Plants do that at night. And so when we took leaves at night and actually measured how much CO2 and oxygen was coming in and out, we were getting more CO2 getting released, and we're getting more oxygen being taken in when the plants were going at high CO2. So they were using this pathway more. Now, you might be like, well, why? Where does that fit into things? And so this is just to remind you that the canopy is busy doing photosynthesis and making carbon. And we've got leaves up at the top of the plant that are doing photosynthesis that we refer to as source leaves. They're making carbon for the rest of the plant to play with. But lots of other parts of the plant aren't either not capable of that process because they're, say, roots, no photosynthesis going on there, or it's a young leaf that's up at the top of the canopy and hasn't yet matured to the point where it can make its own sugar. And so that source leaf that takes in the CO2 is then exporting its sugar products to the rest of the plant in order to allow growth. And so if you do more photosynthesis, you should be able to do more growth. But the respiratory step in this process is really important because it actually requires energy to export. Just like manufacturing, if you have a factory and you increase the rate at which you can make cars, if you can't put those cars on transporters and get them to the showrooms, you aren't selling any more cars, right? They're just sitting in the parking lot of the factory. And so that's where respiration comes in. You can think of it as providing the energy for that export process. Another way I like to think of this is you can think of the carbon budget of the plant as being a little bit like the cash flow of a company. So photosynthesis brings in the carbon. Respiration burns some of that carbon in order to make energy and in order to build molecules. But you lose some carbon in the form of CO2 in the process. And so you can basically calculate how much a plant weighs by knowing how much photosynthesis it did and how much respiration it did and taking one away from the other. And then if you're a farmer, or you're in the business of worrying about food, yield is just the portion of the biomass that was left at the end that you actually get to keep. And so you can think of that as being like a company where photosynthesis is revenue, respiration is expenses, biomass is profit, and yield is the dividend you pay to your shareholders. And so respiration is kind of a confusing thing to think about because you might initially think, well, if respiration is going up, that's bad because the plant's just like breathing all that CO2 back out into the atmosphere that it just worked really hard to get in photosynthesis. But it's generating energy as a product of that, and that energy is allowing it to do stuff. So if you're running a company, you don't want to flagrantly increase your expenses by taking everyone to Hawaii for a company meeting every 2 weeks. That would be wasteful. But if you've got expenses related to, say, research and development that might improve the efficiency of your company, you're getting something out of that. And I would argue here the increase in respiration is a good investment in expenses because it's actually letting the plant get that extra sugar out to the rest of the plant. I like to use Stewie to demonstrate this point too. So, if Stewie is eating his spaghetti on the left-hand side, we've gone from soybeans to Stewie. If this is ambient CO2 Stewie over here, and he has a small plate of pasta, he doesn't have to use all that much energy to get the fork into his mouth to feed himself. Elevated CO2 Stewie, if he's got a big plate of pasta, if he doesn't use extra energy to fork it in, he's not going to get bigger, right? So he's got to turn that fork over more quickly in the same period of time in order to actually benefit from having a bigger plate of pasta in the first place. And so I think this is that's exactly the same idea. In high CO2, the plants effectively have a bigger plate of sugars sitting there in the leaves, but what they've done is they've adjusted themselves, and we can see the gene expression patterns that have allowed it to do that. They've adjusted themselves in order to be able to get that extra sugar out to the rest of the plant, which if they're going to make extra seeds is absolutely essential. By coincidence, we discovered that other people who were looking at plants that are grown high CO2, they'd gone in and counted the number of mitochondria in leaves of plants at high CO2, and they'd found out that in lots of species, including soybean, there were quite a lot more mitochondria in the leaves of plants grown at high CO2. And so that sort of fits with our story. If you think of the gene expression responses that we're reporting as being the plant making more respiratory machinery, this is like them having aerial photographs to say, oh look, there's more factories that machinery is going in. So that sort of supports it. This is a nice example of where genomic technology actually helped resolve a good scientific argument. So my older colleagues had spent about 20 years arguing about which way respiration went, one way or the other. And they were doing that by just measuring those respiratory fluxes, and they kept getting different answers. And some of them thought it went up, some of them thought it went down, some of them thought it didn't change at all. But having this extra layer of mechanistic information helped us disentangle what was actually going on. And we've done a whole bunch of follow-up experiments, which I'm not going to tell you about because I'm already over time, which help us distinguish between these different responses.
The other thing, I think I'll skip this because I'm running over time. The other key thing I wanted to get to was just, okay, we learnt that, great. How can we actually use that in a helpful sort of way? The other thing was we really want to understand those genes that are the master switches controlling all of this. And so we had information on that. So those genes were also in our data set. And so we just went and looked at which genes changed in 2006 and which genes changed in 2005, and then plotted them on this XY plot. So if all of the genes, remember I said there were between 2,000 or 3,000 of them? If all of them came back perfectly identically in every year, we would have had an exact straight line. And some of them show greater expression. So the ones that say are for respiration would be in this cloud because that's a positive response in both years, would put you in this quadrant. There's a cluster of them that go down, which are negative in both years. And then we've got what I call the flip-floppers, who can't make up their mind. These are almost certainly statistical noise. So we're just sort of ignoring them. But we got about 700 of them that fell pretty close to this 1-to-1 line. And so these are the genes we're most confident really, really respond to high CO2. And so we then went in and said, well, what do they do? You know, how many of them have functions that are consistent with being master regulators? And we found a bunch, about 25 transcription factors in particular. And so we then went back. Are you guys familiar with Arabidopsis? Yeah. And so, you know, this is the sort of workhorse lab rat of plant biology. And it's really cool. If you have a list of genes you're interested in, you can just go to a website and order seeds that have individual genes knocked out. So if I had those list of 25 transcription factors, I could just send one of my graduate students to a web page and they could basically just have 25 envelopes of seeds mailed to them the next week with plants that already have those particular genes missing. And so if we want to understand what those genes do, having a plant that has that gene missing and comparing it to what we call a wild type, you know, that has the full suite of genes, that's a really nice initial experiment to do. And so basically what we did is we grew those plants in those cabinets I was talking about upstairs, and we looked at how big they grew. And so here's the wild type, and the black again is elevated CO2. You can see they grew bigger in high CO2 for all the reasons we've talked about. But when we looked at these 3 transcription factors, which in the soybean we had seen respond to high CO2, when we knocked those out, high CO2 stopped being of any benefit to these plants, which we were pretty excited about. And so we're just now starting to do more experiments to see if we can work out what these guys do and why this might be the case. And so this is academically exciting right now because they're not responding and it suggests they might be important. It's not practically useful yet because for it to be practically useful, we would need to know whether turning these things up actually made the benefit greater. So we're quite a long way away from that, but we've taken a step in that direction. And I can tell you that these things, at least 2 of them, are involved in changing plant hormones. And those hormones are probably really important in altering the way the plants grow.
On the last 2, under the normal conditions.
Under ambience, they do a little better under ambient CO2, yeah.
So is it just that you're maxing out?
Possibly, yeah. And so that's the experiments we're trying to do now. Yeah, these guys may not be interesting for that reason, but we don't know. So just to summarize, one point I wanted to leave you with was the idea that these sorts of genomic tools, those are experiments you can actually go and do in the outside world of the field. It's not just a sort of white lab coat stuck indoors scenario. And this whole change in gene expression I've been describing we refer to as transcriptional reprogramming. So it's the reprogramming of the transcriptional patterns that determine how the plant performs. And that's really important in allowing these plants to adjust to perform better at high CO2. And we think now we're starting to get to the point where we understand or we have a list of candidates for what some of the switches might be that turn that response on and off. And if we can manipulate that, we could potentially produce plants that do better in high CO2. And that's really it. I'm talking about it, but a whole list of people were involved in actually doing that work, and it was funded by the Department of Energy and the National Science Foundation primarily.